professor, physician, epidemiologist. I am Dr. Sri Banerjee.
Hello everyone, I'm Dr. Sri Banerjee, core faculty for the College of Health Sciences and Public Policy at Walden University with the joint faculty appointment at Ross University School of Medicine. And in this video, what I wanted to talk about and go over is really understanding big data, but also in the context of medicine, specifically precision medicine. So how does public health, population health, and big data kind of overlay on precision medicine? So, what are some of the sources of data? If we think about the sources first, then this contextualizes this much better. So the areas that have seen a lot of growth are high throughput next generation genomic, metabolomic, transcriptomic, and proteomic arrays coupled with widespread adaptation of the electronic health record. So this is what caused an era of big data in cardiovascular disease investigation. So what I wanted to say as an aside is that a number of years ago, I attended, actually in Philadelphia, this was a conference, and I was introduced, interestingly enough, I actually presented a conference paper and was actually even got an award for it. But in that conference, I was introduced to the concept of omics. And that's an interesting concept because it's an integration when you're adding the word omics or omics into transcription and proteomics. It's actually the interaction and the whole gamut of tools that go around understanding each concept. So it's what I'm trying to say. It's more than just studying a genome. There's conducting associations, demonstrating associations between genomic information and outcomes through the types of clinical information that's available is part of big data, and also actionable data. So it's not enough to have just data analysis for the sake of data analysis. There has to be some purpose, and in this situation, that purpose is to be able to create actionable data. So this is more pertaining to cardiovascular disease, and there's a lot of endophenotypes, intermediate phenotypes, thrombosis and fibrosis that's taking place within the context of cardiovascular disease. So, of course, the first generation of epidemiologic studies, when we go back to Framingham or some of the other studies on a cohort of originally 5,280 people, but that evolved to looking into more and more granular types of information as it is becoming available. So what cardiovascular disease looks like in the era of precision medicine and big data is the fact that there's just so much information specifically in cardiovascular disease and heart failure, and understanding some of these diseases on a specific level in the context of diastolic relaxation or or aging or obese populations, how all of these comorbidities come together to create this disease. This is what big data heralds into the new era. This is what we are trying to integrate in understanding medicine. But before we do that, before we can even move forward and understand cardiovascular disease in this new context, we must first deconstruct genotype as a determinant of cardiovascular phenotype. What does that mean? And this is a debate, nature versus nurture, and many different types of debates that we've heard of previously. But here, the idea is that the common viewpoint is that you have a heritable component for a limited number of types of heritable components. And that has fueled the search for pathogenic genes to implicate as causal factors. Now, that couldn't be further from the truth. The truth is that there may be some relationship, of course, between, for instance, the low density lipoprotein receptor gene or the familial hypercholesterolemia gene. And so there are genes, specific genes that does accelerate atherosclerosis, and single genes that does indicate heart disease. We can't deny that fact. But in this neuro model, we have to also look for protein inactivating variants, or ones that also lead to improved outcomes. So one of them is a protein inactivating variant, which is NPC1L1. And then patients treated with anti-NPC 1L1 pharmacotherapy actually are helpful, and also the loss of function mutation in ANGPTL4. These are associated with lower triglyceride levels and protection from cardiovascular disease. But we have to, even this, even though this is another approach, what we need to keep looking for is another model where there's a complex genotype-phenotype relationship. This original model is implausible for most cardiovascular diseases. So what does this say? One deletion or one genetic determinant is not enough to be looking for cardiovascular disease. We have to go deeper and understand the endophenotype. And that's a critical step in the development of clinical disorders. So the expression of overt cardiovascular and pathophenotypes, rather than focusing on that, it's important for us to focus on myocardial infarction as and I like the way this is worded, so I'm gonna read what it says. Convergence of perturbations and numerous potentially phenotypically related genes that are subject to further modification by an individual's exposome. The cumulative environmental exposures that affect health over an individual's lifespan. So this is going over and understanding how an MI, a myocardial infarction, which is an acute event, and I tell this to new students and people that are doing research in myocardial infarction, is that even though you might not think about this concept, there is a difference when you're thinking about chronic diseases like cardiovascular disease. There is a difference between acute versus chronic. So for instance, myocardial infarction is an acute indication of heart disease. Now, a chronic, very common indication of heart disease is congestive heart failure that can evolve over years. So it's a complex convergence of perturbations. It's an interesting confluence of all of these that leads to the cumulative exposures. Now, in a genome-wide association study, in a large scale, this was a strategy that was also used in the last 2 decades, and provided us with a lot of information as to, for instance, there was a meta-analysis that I want to highlight: 184,305 individuals with MI and reference. This was the Exome Aggregation Consortium, further establishing that individuals carried 54 mutations considered pathogenic, but approximately 41 of these mutations occurred with high enough frequency in the general population that they were considered unlikely to be causal for several diseases. So that means that there were only 13 mutations out of the 54 that actually made it to show that these were causing disease. So although this is a good model, it's important to make the transition into big data and precision medicine. Instead of looking at genes, maybe the existence of resilience genes or gene modifiers may be an important factor as well. For instance, in diseases like pulmonary arterial hypertension or hypertrophic cardiomyopathy, these are all heritable. But these disease-causing mutations do not manifest in the disease phenotype. So there's actually disconnects. So even if there's a phenotype, doesn't mean that you'll see it in the genotype necessarily, or we don't have mechanisms to be able to see it. So looking at a cohort of 55,685 participants, individuals with a higher genetic risk score subscribed to three of four healthy lifestyle factors, and had 46% reduction in relative risk for coronary events. So this is definitely an effective gene score because it's taking into consideration and taking into account multiple factors to be able to explain cardiovascular disease. Again, these are all applications of big data in different contexts. And have you seen how the transition is made between classical genetics, where originally there was Mendelian genetics, where there was classical inheritance patterns, and then of course genome-wide association sequencing, which is a little bit more complex than some of the deletions or simple mutations that I explained about with the connection with cardiovascular disease. And then talking about in a large meta-analysis about out of a total of 54 mutations, only 13 explaining myocardial infarction and cardiovascular disease. And then going into understanding how big data has really the opportunity to provide enhanced clarity between genomics and precision phenotyping by increasing sample size, enhancing the characterization of more nuanced pathophenotypes, providing data important for understanding gene modifications, and offering unique analytical strategies heretofore unavailable, chief among which is the application of network medicine to complex, heterogeneous molecular interactions. what I like about this article is that it's combining, it not only a major chronic disease and the application of big data, but also it's actually integrating molecular epidemiology, which is a concept often neglected in some of the core epidemiological curricula that we often talk about. So, talking from transitioning now from deconstructing genotype as the determinant of cardiovascular phenotype to deconstructing clinical cardiovascular phenotypes. What does that mean? So we start off by saying most cardiovascular diseases are not circumspect entities that involve a single cell type or organ system in isolation. So we need to scratch that thought first, first and foremost. It's important to understand that it's not a single cell that is causing disease, and deconstructing that has important implications in the applicability of a conventional reductionist model in informing an understanding of cardiovascular disease pathogenesis and phenotype. So the use of a reductionist model has been has been kind of thrown out the window, and we're transitioning into big data and the connections between genotype, endophenotype, and then clinical phenotype. And there's actually even an acronym for this, GECP. So the simplistic GECP is what illustrates the dilemma that big data analytics must address. These connections before, and I've been a part of my specialty 20 years ago was in molecular biology. So at the time, a lot of these a lot of these concepts were at its infancy. So transitioning from kind of those concepts, and of course I took genetics as well and advanced concepts in genetics. Transitioning from these understandings to where these connect and these complex perturbations connect to create chronic diseases is an interesting growth and transition to observe. And so some of these conditions, acute and chronic, as you'll see, involves the applicability of big data. And so myocardial infarction and ischemic heart disease, these are both acute, late, and chronic phases. They're considered of chronic phases of myocardial infarction. Now, again, I took cell biology. Of course, this was something that was considered, and then, of course, in pathophysiology, in histology, looking through the microscopes, looking at different cells. So we can't discount the fact, although this is reductionist, we cannot discount the fact completely that that's an approach that must be looked at before deconstructing. So when we're looking at crosstalk, and this is a concept that's oftentimes used in cell biology, between cardiomyocytes and fibroblasts, endothelial cells, and circulating immune cells, like monocytes. Monocytes, by the way, are precursors to macrophages. So monocytes and T lymphocytes. So when you're looking at this is just an aside, but when you're looking under a microscope and when you see a lot of monocytes, that means that there's a lot of loss of white blood cells, and there's something going on which you need to be aware of within the body, which you need to be looking into. Anyway, so back to the concepts and combining them. So you have this crosstalk between these cellular levels. And then there's pathophysiological changes that takes place. For instance, in ischemic cardiomyopathy. What is that? Ischemic means starving of blood. And when you starve the blood supply, also you are starving oxygen, which is being carried by hemoglobin. And then you're also starving the area of nutrients that need to be traveling between the bloodstream into the cellular level. So these concepts and things do not change. You have to be still thinking about systolic differences in systolic function before an MI and after an MI, what happens in myocardial remodeling. And this is a very complex process that if you are a medical student, you have to know very well for your USMLE exams. And also looking for mechanical complications across different patients and again temporally within the same patient. So in medical research, and now that I'm teaching in that context as well, a lot of times case reports of a single patient looking through time, it can provide a lot of insight to other physicians due to the fact that almost each patient, you can say, is unique and has unique characteristics. So you're trying to apply research which is applying to the population to one individual patient. So there's not a lot of experience in treating that one patient because there is no other patient that is like that one. So when you're using and applying population health framework to this, you have to get a little bit more granular in the types of information that you have. So I would argue that big data is how you can get more precise information and lead to precision medicine. But then there's actually a next step after you have your data that has been collected for precision medicine. Because then, now that you have further categorization of patients, more precise categorization, different categories than you originally started with, but more precise categories, now you can have more precise treatments exactly where it's needed. And you can take into consideration different genetic types of information in understanding the potential for later development of disease. So this is not only looking at myocardial infarction, but hypertrophic cardiomyopathy. And a lot of this is from epidemiological concerns of cardiovascular disease being the top leading cause of mortality within the United States. And this is every year, year after year, this is the fact. And so about, if I recall, about 700,000 individuals or in that magnitude each year pass away from cardiovascular disease. So many of my studies actually look at cardiovascular mortality, and honestly, a lot more needs to be done in this area. And I think the idea is that this will lead us and help us leap. Big data will help us leap into a new era and allow us to bridge the gap and allow us to understand how to integrate how high throughput information. And these diagrams provide much more insight. And I want to close this up as well and not go into too much detail. But this is an interesting figure to be looking at. Big data enhances precision cardiovascular phenotyping. So what types of, you might be thinking to yourself, what types of big data sources am I talking about? Well, panomics, everything, the microbiome, so what type of microbes are within, proteomics, metabolome, what are your, for instance, your heart rate, respiratory rate, for instance, your oxygen levels, what else? Anything else? Transcriptome, and then genome. And then you have network analysis, big data networks, and I'm going to get into the network of networks. But you also have access to all of this clinical data that you never had before. Not that you ever, you did not have this information, but never before was all of this information streamlined and integrated into set of um, one set of data. So you can look at blood work, imaging, physician notes, prescriptions, fitness devices, like a Fitbit. So all of this information can be integrated and then create another big data network. So these sources can then create big data network and then expose them. What are is importantly environmental, social, but also some dietary and habit information. So these can be known as big data networks. And then these big data networks come together to provide a network of networks. And then when you start conducting an analysis, then you can either have data from populations that can identify clusters of similar phenotypes within a heterogeneous pathophenotype. And this was what I was explaining about creating new clustered phenotypes and heterogeneous pathophenotypes, which then allow you to meet patients where they are and be able to then treat according to what the specific needs are in each group. So transitioning into understanding some studies about hypertension, such a common and in some ways underdiagnosed condition that it's important to look at. And there's so much heterogeneity in the phenotype of hypertension and dysregulation of vascular structure and function attributable to endothelial dysfunction. These are all, there's there's a lot of heterogeneity on all this. There's variations. For instance, increased vascular smooth muscle stiffness, aberrant matrix production by adventitial fibroblasts and resident progenitor cells, and inflammatory immune cell infiltration of vessels. So all of these, they might be, they might sound like a foreign language to you, but the idea is that in all of these areas, there's so much, so many differences, and also that hypertension in and of itself is provoked by physiological stress, neurohormonal regulators, salt intake, obesity, um, which may occur together or separately. But um, efforts to identify polygenic basis for hypertension have identified variants associated with the pathophenotypes. And of course, this is studying from the initial Framingham heart study, which has now evolved into a genotypic consideration. So 33 SNPs, single nucleotide polymorphisms, were related to blood pressure or hypertension. And this demonstrates that the prevalence of hypertension increased commensurate with genetic risk. And so this confirms 34,433 individuals with genome-wide significance. Similarly, there were about 140,886 individuals of European ancestry in the UK biobank. And then when a meta-analysis was done over 1 million individuals, it was found that 535 new loci were linked to blood pressure. Multiple studies have been done over the past years and have indicated different places which lead to cardiovascular disease. So let's look at the expression of phenotypic data. We talked about precision cardiovascular phenotyping, but now talking about how the classic model and then the contemporary model, where the differences are. So before, the idea was, here's the disease, and we do a genome-wide association sequencing, and then look at the single gene variant, causes complex cardiovascular disease phenotypes of inflammation, thrombosis, calcification, fibrosis. This is still a good place to start, but we have to be thinking a bit more about different sources of heterogeneity. So where are areas where there are variants? So the LDLR is a variant, and that is abnormal LDL metabolism. And when you have abnormal, that's bad cholesterol in lay terms. When you have that, you have more thickening and angina, which is pain within the heart, myocardial infarction, left ventricular remodeling. Now that takes place when the ventricle starts to get damaged, and then heart failure, and then VT substrate. And then transitioning to pulmonary arterial hypertension. There's the BMPR2 variant, and then there's smooth muscle proliferation, vascular smooth muscle proliferation, and then vascular fibrosis, apoptosis, resistance, dysregulated metabolism, and then right ventricular adaptation. Notice we're talking about right ventricle because this is something that is considered more of increased pressures within your pulmonary artery. And if you remember your cardiovascular physiology and pathophysiology, then this will make sense. But just going through the blood circulation within your cardiovascular system, if you think about the superior and inferior vena cava emptying into your right atrium and then going through the valve into the right ventricle, then the pulmonary artery over there actually takes away deoxygenated blood that has returned to your heart and takes it to your pulmonary artery through the right ventricle. When the blood goes into the pulmonary artery, the pressures in there need to be a certain point, or else the blood will not sufficiently flow, and then back up into the right atrium and superior and inferior vena cava, which could then lead to ascites within the liver, and then also inflammation within the leg. And so the blood continues to circulate into the pulmonary, into the capillaries, where there's gas exchange between the lungs. Within the capillaries, the cell lining is the endothelial cells are very thin, so that there can be exchange. And then after that, the pulmonary veins carry oxygenated blood into the left atrium and going through the mitral valve goes into the left ventricle, through which the blood is carried into the aorta, and then into the axillary artery, which carries blood into the axilla, and then the brachiocephalic artery, and then the common carotid. So these are arteries that are going off of the aorta, but one of the first arteries that come off of that is the coronary artery, and so this is supplying actual blood to the heart. So the blood is so busy, so the heart is so busy pumping blood to the rest of the body that it almost like forgets to feed itself. And so these nutrients need to get to the heart. Um, so this is explanation of pathophysiology of pulmonary arterial hypertension. And then cardiomyopathies. Tragically, some of the cardiomyopathies can affect people. I've actually known individuals that in their teen years passed away due to an unknown cardiomyopathy. And then when they went to play sports, their heart was strained beyond the capacity that they could use, and unfortunately passed away. So this can actually offer hope for individuals. The TTN variant for preventing heart failure and dilated cardiomyopathies. So that's another example of big data. But then the contemporary model looks at, and I'm going to quickly go over this, the biological risk and the possible acquired factors. So genetic, transcriptional, post-transcriptional, and epigenetic, which is actually an interaction between the environment and genes, which takes place after the genes have been expressed. So these are like DNA methylation patterns and others which lead to epigenetics. And then infection. Of course, infection not only has, as I've illustrated through many types of research, that infection doesn't just affect short-term sort of acute factors, but also goes into chronicity and even cancer. And then it depends on exposures, also different toxins, and there's a lot of information and big data that is associated with that. And then there's other sort of possible acquired factors that we can be thinking about. So the combination of biological risk and the possible acquired factors lead to gene variants. The endophenotype and the endophenotype 2 lead to endophenotype 3. So then here is a combination of many of the chromosomal data, the pathogenic gene variant, the endophenotype, biochemical and physiological, and then pathophenotype and treatment. So myocardial infarction and medical therapy. So how does this work in terms of individuals? I've talked about population levels, and some of the sources of phenotype heterogeneity in cardiovascular disease in clinical trials can be seen in the CHARGE Consortium, Sweetheart, and the UK Biobank Initiative. And then also the NIH All of Us study, and then Million Veterans Program. So check into these for big data sources, and looking for relationships with hypertension, coronary heart disease, and MI. And this diagram goes into big data informing reticular typing and cardiovascular disease phenotyping. And then this ends with some of the opportunities that are available, but also limitations. Of course, we've talked about how big data can lead to much more diverse longitudinal data sets that inform cardiovascular disease and incorporate many levels of data such as laboratory imaging, biological, and descriptive data with heterogeneity in data collection frequency. So the electronic health records is already primed for data mining. And despite this wealth of information, the EHR lacks standardization. So there's a need for a global universal language for big data. And so physiological, for instance, sleep parameters, nutritional data, these are pivotal for acquiring cues, informing individualized cardiovascular profiles. I hope this has provided you with thorough, I should say, overview of network medicine, precision medicine, the role of big data, and how this can really provide revolutionary type of medicine, which focuses on individual patients and individual groups of patients rather than looking at this from a population perspective. Thank you for listening.